Dear fellow researchers, hello everyone. Welcome to this technical cup. Today we focus on life science research, protein quantitative technology for one of the core basic technologies, protein as the primary performer of life activity. Its content and functional state are directly related to cell physiology. Key processes such as developing cases in proteinomatic analysis, analysis of protein structure and point verification in research scenarios. Precise protein quantity is to ensure the reliability of experimental data. Repetitive premises. Currently prevalent protein quantitative techniques can be divided into ultraviolet spectrometry, pen coloring. The ultraviolet radiation method is based on the aromatic amino acid marameate, chromium chloric acid. Characteristics of absorbing wind at 280 nanometers wavelengths. Concentrations are calculated by the law of Lombardia. This method does not require labeling samples that can be recycled. Suitable for high-purity protein, fast quantitative, but susceptible to interference with ultra-absorbing substances such as nucleic acid corrosive acids. Requires written quality correction through the A260 and B220. Pen color is the preferred choice for laboratory-regulated quantities. This includes the BCA law, Bradford Act, and Lowry law. BCA is used in alkaline conditions. Proteins' effect on the restoration of ketone ions. The acetone ion produced with the BCA formula forms 562 nanometers. With the exception of purple, falls with maximum absorption. Its dynamic range is wide 20 to 2000 micrograms per milliliter. Compatible with most pollutants. Bradford's law is based on the code of thinking G250. Combined reaction with protein alkaline amino acid disability. Detection of light absorption at 595 nanometers. Operation simplify reaction fast. But it is more sensitive to disrespect and only suitable for proteins with a molecular mass greater than 3 kD. Fluorescent quantitative techniques such as the Cuban protein quantification method. By combining fluorescent dyes with the specificity of proteins. Using the linear relationship of fluorescent intensity and concentration to reach quantitative. Sensitivity can be given at the Danic level, suitable for microsample testing, and its resistance to interference is significantly superior than that of the color method. Nature spectrum technology provides high flow for proteinomy. Quantitative programs with high accuracy. Includes TMT-SILAC technology based on isotope marking. And unlabeled label-free quantities. These methods can not only reach absolute quantity, it is also possible to simultaneously analyze the expression levels of several thousand proteins. Screening of disease markers. Drugs play a key role in the study of mechanisms. When choosing a quantitative method, comprehensive consideration should be taken of the sample concentrations, net flow demand and experimental cost. For example, thyroid protein samples that do not contain restaurants. Priority choose BCA law. Adamus Life's BCA protein quantitative reactor box. Quality stable, widely appreciated. It is your ideal choice. For thyroid protein samples containing restaurants, can use Adamus Life's Bradford protein quantitative reactor box. Microcomplex samples recommend quantitative fluorescence or spectral technology. Rapid filtering can be done using ultraviolet spectrometry. Quantitative accuracy is the cornerstone of scientific exploration. Will provide a solid data support for your research. For more information, welcome you to the Exploration platform, or consult a local dealer.